Microbial study of Borassus flabellifer L in the conservation and preservation of palm leaves Tala Patra manuscripts. Ashwini M. and Ramakrishna Bhatke. Aim. The aim of the study is to access the microbial activity of Tala Patra leaves palm leaves manuscripts PLMB flabellifer for the preservation and conservation of Tala Patra manuscripts. Study design. The study is designed in such a way that the leaves sample were used for the different microbial study in the science laboratory. The observation was recorded and the result is predicted with the help of the standards. Place and duration of the study. The leaves sample of Tala Patra was collected in the Department of Manuscriptology, Karnataka Sanskrit University, Chamarajpet, Bangalore. The work has started in February 2024. Methodology. In this work we have chosen microbial isolation was done from the leaves sample collected by using suitable growth media under controlled condition to facilitate their growth and study. Results. This study possesses the antimicrobial activity which will inhibit the growth of various microorganisms, bacteria, fungi and even viruses. The study supports us to know the leaf microbe interactions, as few microbes have symbiotic association with the plant and few could be pathogenic to plant. Conclusion. According to Constitution of India, 1950, in Part 4A under Fundamental Duties in Article 51A, it shall be the duty of every citizen of India. No. F. To value and preserve the rich heritage of our composite culture. Government of India 2015. Therefore, it's everyone's duty is to preserve the heritage of India by preserving the PLM. In this context, microbial studies of B. flabellifer manuscripts leaves offer valuable insights into the potential on the manuscripts and provide a basis for the preservation and conservation of original PLM. Thank you for watching this video. Please subscribe to our channel to watch more scientific videos.